Hi, my name is Ethan Guo. In this video, I want to talk about the simulation work that are done at the University of Rochester about tiny valves in the brain. Valves and valve-like mechanisms exist in many biological systems. Here I'm showing some examples in human body. Heart valves act like one-way doors that keep blood moving forward through your heart and stop it from flowing backward. Similarly, the vein valve keep the blood flow in the veins in one direction, and dysfunctions of these valves might cause diseases. Another interesting valve mechanism is in the lymphatic vessels. There are valves in the collecting vessels allowing flow in one direction. And there are also these tiny valves at the lymphatic capillaries. These valves are formed by neighboring cells and they flap open and close to only allow fluid flows into the lymphatic vessels. And here's a recent imaging study showing the open and close motion of these tiny valves. We think similar valve mechanism might also exist in the glymphatic system in the brain, and we did numerical simulations to study it. So what is glymphatic system? There are spaces and channels in the brain that are filled with fluid, called the cerebrospinal fluid. The glymphatic system uses the fluid that flows through the spaces and channels. This animation shows a close-up view of the glymphatic system near the brain surface. There are perivascular spaces around blood vessels formed by supportive glial cells called astrocytes. The fluid flows through the perivascular spaces around the artery, then flows through the spaces in the brain tissue, then enters the perivascular space around the vein, and then goes to the lymphatic vessels. In this process, the metabolic waste proteins produced in the brain are taken away. However, how the fluid flows across the wall formed by these astrocyte end feet is not fully understood. We used the imaging data to reconstruct a 3D numerical model to study it. As you can see from this microscopic image, the astrocyte end feet of different sizes surround the blood vessel in the brain. And in our simulations, the pressure difference between the perivascular space and the brain tissue side changes periodically from -5 to 5 pascals. When the pressure is higher in the perivascular space, the end feet are pushed outward, and the gaps between the end feet open, which allows fluid to flow out. However, when the pressure is lower in the perivascular space, the end feet are pushed inward. But due to the cylindrical geometry of the end feet arrangement, the gaps are closed. And the plot shows the volumetric flow rate, which increases as the pressure is higher in the perivascular space, allowing the fluid flowing out of the perivascular space, but almost no fluid flows in. This is mainly due to the curved shape of the end feet, and to quantify how the curvature can rectify flow, we did simulations in 2D with 2 end feet, and in 3D with 3 end feet. Both cases show that this valve mechanism due to curvature can act like one-way door for fluid flow. The microscopic image also shows neighboring end feet could have different sizes, so another hypothesis we have is a longer end foot overlapping a shorter end foot. Similarly, we did 2D and 3D simulations. The longer end foot deforms more and opens the gap when the pressure is higher in the perivascular space and closes the gap when the pressure is higher in the brain tissue side. The flow rate plot shows this mechanism can also rectify flow. There is another hypothesis about the gap being wedge-shaped, and we ran simulations to verify that this wedge-shaped gap is also very effective in turning periodic pressure conditions into one-way flow, as shown in the animations and the flow rate plot. This work is published in the Journal of Royal Society Interface. If you want to learn more about it, here are the links to the paper and to our lab's website. Thank you for watching, and I also want to thank NIH for supporting this work.